Protein synthesis. The two steps in protein synthesis are transcription and translation. In transcription, mRNA is synthesized based on the DNA template of a gene. This is followed by translation, which involves the production of a protein with an amino acid sequence that is based on the nucleic acid sequence of the mRNA. The central dogma of molecular biology stated that genetic information flows from DNA to RNA to protein. The translation nucleotide sequence to amino acid sequence uses the genetic code. Transcription. Transcription occurs in the nucleus of the cell. It is the process of copying sequence of DNA to mRNA. There are three defined states in the transcription process: initiation, elongation, and termination. Initiation. For each gene, only one strand of the double-stranded DNA molecule is transcribed. This strand is called the antisense strand or template strand. The other strand, which is not transcribed, is called the sense strand or coding strand. It has the same sequence as the product mRNA, with thymine instead of uracil. Elongation. During the elongation phase shown, the RNA polymerase complex works its way along the DNA molecule, synthesizing a strand of mRNA that is complementary to the template strand of DNA in 5 to 3 direction. Termination. When the RNA polymerase complexes reach the termination signal, they detach from the DNA strand. The new mRNA strand is released from the transcription assembly. Modification of mRNA. The newly synthesized mRNA is called precursor mRNA or pre-mRNA. Before moving out from the nucleus to cytoplasm, the following modifications convert pre-mRNA to mature mRNA. 1. Addition of a 5 cap at 5 end. 2. Addition of a 3 poly A tail at 3 end. 3. Removal of introns, non-coding regions, with specific enzyme and the remaining exons, coding regions, are joined. Translation. The second stage of protein synthesis involves translation, which occurs in the cytoplasm. It is the conversion of a sequence of nucleotides into a sequence of amino acids in the polypeptide chain. There are two subunits in the ribosome. At the small subunit, mature mRNA is attached. There are 3 binding sites for tRNA on the large subunit of ribosome: A site, P site, and E site. Each tRNA carries the specific amino acid at a time at acceptor stem. Opposite to the acceptor stem is the anticodon, which connects to the specific codon on the mRNA. Initiation. In the initiation phase, all the translation components come together. The small ribosomal subunit attaches to the mRNA near the start codon, AUG. The first tRNA that binds to the codon at the initiator tRNA with its UAC anticodon, which joins the AUG codon of mRNA. Then, a large ribosomal subunit joins to form the active ribosome. Elongation. The polypeptide becomes longer, adding 1 amino acid at a time. During elongation, a cycle of 4 steps is rapidly repeated. First, a tRNA with an attached polypeptide is in the P site, then peptide bond is formed and tRNA carrying the next amino acid enters the A site. Next, the polypeptide chain is transferred to the amino acid of the tRNA in the A site. This makes the polypeptide chain 1 amino acid longer than before. Last, the mRNA moves forward by 1 codon, and the polypeptide-bearing tRNA is now at the ribosome P site. The uncharged tRNA exits. The new codon is at the A site and can receive the next complementary tRNA carrying the next amino acid of the polypeptide. Termination. The termination phase begins when a stop codon on the mRNA is reached. The polypeptide and the components of the translation machinery are separated. A protein, called a release factor, cleaves, cuts the polypeptide from the last tRNA. The polypeptide is released and will eventually fold into its 3-dimensional shape as protein, ready to carry out its cellular activities.